Dr. Ong Sin Tiong is an associate professor in the Cancer and Stem Cell Biology Signature Research Program and a visiting consultant at the National Cancer Centre and the Department of Haematology, Singapore General Hospital. Dr. Ong graduated with a medical degree from Cambridge University in the UK and completed his clinical and subspecialty training in medical oncology and haematology at Cambridge University, the National University Hospital, Singapore, and the University of Chicago. Prior to returning home to join the Duke-NUS Graduate Medical School, he was on the faculty at the University of California at Irvine, where he directed a research group focused on understanding drug resistance in leukemia. Recent work from his lab has identified genetic reasons why East Asians are less responsive to certain cancer-targeted therapies and how novel drug combinations may overcome this particular form of resistance. In addition, his lab is collaborating with the Experimental Therapeutics Center, A*STAR, on designing new drugs to overcome a particularly deadly form of human leukemia called blast crisis chronic myeloid leukemia. At what point in your career did you choose to become an investigator, and why?
What I realized as a clinician seeing patients was that the aspect that I really enjoyed most was helping people. And the people that come and see us are those with cancer. And so whatever we did was doing something that I felt was very meaningful, because these are obviously deadly diseases. But what I realized over time was that if we could solve their important problems in cancer at a mechanistic level, our discoveries could potentially affect many such individuals. And so that if you make an important discovery to do with drug resistance or identifying a new target in cancer, are able to translate that into a new treatment, that could potentially affect hundreds, thousands, tens of thousands of individuals. So that realization, as I progressed in my career, I think was what made me switch my focus and my career direction to becoming a clinician scientist or someone engaged in translational work.
At the point you made your decision, how adequate did you feel?
I think I still felt totally inadequate, because I felt I didn't have the complete skill set. I didn't have much in the way of resources, and I didn't have a single mentor whom I could model myself after. So, and furthermore, after a couple of years in my first job, my project completely failed, so it totally bombed. And as a result, I was left with very little at that point. So it was a very depressing time of my life.
What were the challenges that you faced in the earlier part of your career?
I think they were related to my being completely inadequate in terms of experience, lack of mentors, lack of funds. But over a series of a couple of years, we were able to rectify the situation. We were able to find a new project which came in a totally unexpected manner, and were able to generate data that other people found interesting, that we were able to use that data to publish, to get funding, and to find interest in the community that we were working in.
What gives you the buzz to continue what you're doing?
I think it is making discoveries that are surprising, interesting, and that ultimately can make a difference in patients' lives. So the best day of the week for me is Thursday mornings, because that's the day when our lab has its weekly meeting, and when I get to hear about what interesting things the people in my lab have uncovered that last week or the last couple of weeks. So that's what keeps me excited. And it doesn't happen a lot, but when it happens, it's enough to keep me going.
Could you tell me more about your mentors?
Sure. So I've actually had a series of mentors over my career, and I think probably my first real mentor was a man called Professor John Altman. So he was a world expert in lymphoma, and I went to train under his wing at the University of Chicago. when I did my fellowship. And I learnt two things which I think were very important from him. One was what it takes to become a world expert in anything. And what I saw was the amount of work and single-mindedness he put to it to become a person of that stature. And the second thing I learned from him was what it takes to look after patients really well. And he would give out his hand phone, or in those days, his pager number to patients. And this was a full professor who was in his 60s, still giving out his pager number to patients. And so, to me, that was just a fantastic model of what it takes. Someone who's really good in terms of the clinical science, but someone who has a real heart and passion for looking after patients so well. So that's one of my first mentors. The other mentor that I had while doing my fellowship was a lady called Michelle Lebeau, who started off as a cytogeneticist, but who's now the director of the Cancer Center at the University of Chicago. So what I learned from Michelle was attention to detail in terms of laboratory work, and again, how much it takes to be, again, a world leader in that field. When you, for example, weigh out agarose to set a gel, we'll weigh it to the nearest gram, but she would weigh it to the nearest 0.000 grams. And I got the feeling that she was doing that purposefully so that I would see that. And so what I took from that is that you have to be tremendously precise, not just with your hands, and she showed it intellectually, but with your intellect as well. So that was Michelle. Another mentor that I think was really important in the development of my career was a guy called Professor Hung Fan at the University of California. So I worked with him for several years. But he taught me how to, I think, interpret data quantitatively, and to not just look at the data in face value, but to see what the greater significance of the information is that you've generated, one. And two, he taught me that when you're writing a grant or giving a talk or writing a paper, that you always have to have an intellectual hook, that you needed to grab the attention of whoever's reading or listening to you. And that was very valuable to me. And none of these lessons are things which you pick up in medical school, which is what we are here at Duke-NUS. So I always tell my medical students that you need mentors, you need different sorts of mentors during your career. And these people will teach you the business of doing science, really, which is what I was looking out for.
If you could go back in time, is there anything you would do differently?
There are probably many things that I would do differently now. But I wouldn't be here if I hadn't gone through what I had to go through in order to get to this position. But I think probably the single biggest lesson for me to learn as someone who just has a medical degree and not a PhD is that the whole process or business of doing science is a very different thing from the whole process and business of looking after patients. So there's an entirely different skill set than one needs to accumulate and become expert at in terms of doing science. And there are many categories within the business of doing science that one has to become good at. And I think I would be more intentional in seeking mentors or experiences that would enable me to become good at these things of the business of science more quickly or more efficiently. So running a research group is very much like running a small business or actually your own clinical department, because you have to set the overall direction of the work that's being done. So you have to give the group an overarching idea or hypothesis that they will then split up and work on in their own individual groups or by themselves. You have to convince your stakeholders, i.e. your funding agencies, that what you're doing is important. So you have to be able to sell your work, both orally and in written format. You have to be able to hire people, inspire them, manage them, occasionally let them go as well. And so there's a lot of HR sort of stuff that we do as group leaders. One has to be able to also talk about what you're doing, not just the public, but the community of your peers in your area of interest. So you have to get yourself invited to give talks, keynote lectures locally, but also all over the world to try and publicize your work. And I think to go to the next level as well in any work nowadays in biomedical sciences is you have to be able to initiate, maintain collaborations, because that's what enables your work to get to a higher level. So there's all these aspects of, I think, running a successful research group that really is not taught much in medical schools. So if I were to do it all over again, which is your initial question, I would, categorize each of these areas and then be more intentional at gaining the skills that are necessary for it.
Could you tell me about a project you're particularly excited about?
One of the projects that we're particularly excited about was something that we published last year. And the genesis of the project was actually an observation that we'd made 10 years ago from other people's work, was that, and this has to do with resistance to so-called targeted therapies in cancer. So in some patients in whom we give this treatment, they fail to respond. And yet we're pretty sure that the drug was hitting the right target, yet the cancer cells were not doing what they were supposed to do, which was to die. I'd taken that observation and stored it away mentally because at that time we didn't have the resources to pursue that line of investigation. But when I moved back to Singapore, it was something that we set about trying to understand more vigorously. And I think when we got the results back, what they told us was something very surprising, was that even though you had this cancer that the therapy or so-called magic bullet was targeting directly, what the results told us was that if you were born with a variation in a normal gene that caused cell death, and you inherited it, unfortunately, from one of your parents, and even though we've had the right bullet for this particular cancer you had, that because of this unfortunate inherited variation you have, you were less likely to have a good response. So in the field that we work in, we're always interested in mechanisms of resistance that are intrinsic to the cancer cell. But what we'd uncovered was a mechanism of resistance that instead you'd inherited from your parents. So that was very surprising. And it was also, I think, very, in a way, delightful for us because we figured out exactly what the variation in the gene did in terms of a failure to activate cell death. And as it turned out, a drug company made a drug that mimicked the function of that gene product. So what we could actually do was then take cells with this variant, and then when we added the drug that corrected the function of the gene product, we found that it was able to overcome resistance to the standard therapy. So that was, I think, a very nice story in that we made a discovery that was totally new. And thirdly, it was great because there was something that you could do about it therapeutically. And as it turns out, we made that discovery in leukemia, a rare type of leukemia. But because we work on fundamental mechanisms of resistance, we were quickly able to translate that discovery at the biologic and mechanistic level into other cancers, which are actually very common in this part of the world. The other thing that was interesting to us was that the gene variant that I was talking about actually can only be found in East Asians, so Chinese people, Korean, Japanese people. It's totally non-existent in Caucasians or in Africans. So it was something that was, again, a very nice twist for us because it was very specific to our part of the world. It satisfies all those things that I wanted to try and achieve when I made that career change to go into translational research.